So this is the prep for the final lecture of the class. We're going to finish up looking at limb regeneration. I'll be introducing some additional proteins and factors, just a few. There's a lot more in the textbook, you'll notice, so these are the ones I decided to focus on. And besides the actual signals, one of the major sort of thrusts of this particular lecture is just thinking about positional information. It was one of the core concepts that was introduced right at the beginning of the semester, the idea of having some kind of morphogen gradient or multiple gradients, and cells understanding, quote unquote understanding, having the information about what position they had in the structure, the tissue, the embryo, whatever it is, the neural tube, based on the combination of different signals and different gradients of morphogens, possibly overlapping gradients. Think back to the neurula of the early frog embryo with the BMP and the wind gradients, for example. That leads to very specific, dorsal ventral and anterior posterior information. And so we're just going to look at proximal distal, but if you think back to the multiple things we've done through the semester, just looking at different kinds of gradients and sort of how the different thresholds gives rise to different cell fates and so on, that conceptually that can help you understand what we're going to talk about. I'm not including too many gross pictures of, I'm pretty sure that amputations and regeneration experiments look awful. But instead I have a lot of schematics. So the signals and so on, and the sort of principles, but also just that whole idea of positional information. Okay, and I'm just starting with this little tidbit. It's kind of interesting, is that even after birth, a lot of mammals do retain the ability to regenerate the tips of the digits when they're still young. In the case of humans, this is up to age somewhere between 8 and 10. And so it's just the tips. So if you think about your own hand, if as a 5-year-old somehow a finger was cut sort of above the first knuckle, that there's a really strong chance that can be regenerated. Whereas if it was below the first knuckle or closer to the hand, chances are it would not be able to regenerate. And it turns out, like experimentally, I mean, I'm pretty sure, I don't know that they've necessarily done this with little kids, but it appears that the region where regeneration can occur is the same area where the MSX1 gene is being expressed. So just in the tips of the digits, even after birth, that gene is being expressed a little bit there. Okay, and so just remind yourself what MSX1 does in limb regeneration, what its role is, and that will help you understand why MSX1 is important in this process, and what it has done to allow that regeneration to occur. Now, if you are working with mutants, so there's experimental work where they look at mutants for MSX1 or for BMP or for noggin, and a number of other different proteins, and it's found that in these BMP MSX1 mutants, if you provide MSX1, it's not enough for the whole regeneration process. And BMP is thought to activate MSX1 in the regenerating digits. And so it's possible that BMP allows for regeneration not just through MSX1, but that there are additional targets that are needed for that. But MSX1 is playing a role, and if you remind yourself what it does in regenerating limbs, that will tell you what the exact role of MSX1 would be in a process like this, even even in mammals that don't normally have the ability to regenerate their limbs. So this is a really key concept, which is that if you look at the cut site, either because a predator has, bitten part of a limb, or because it's been very cleanly and sterilely cut, um, the the tissues that are regenerated are always more distal relative to the cut site, so not more proximal. And you know, this had to be studied experimentally, of course, but you would think, like in nature, is there ever going to be a situation where a proximal part of a limb is lost, but not the distal part? Because, right, it's, under those natural conditions, that's certainly not going to happen. So one thing to think about when we're talking about this subject is just, you think about like the natural, when would this have been useful, and why did it develop? If you have high levels of predation and you're losing a limb or a tail, or a little bit of your dorsal crest, what part is most likely to be bitten off? And so therefore, what should you need to regenerate, right? Now there are I will be describing some experiments where things are grafted together that wouldn't normally ever happen in nature, or reversed in orientation and so on. These are experimental manipulations that help us understand better the process and how this process occurs, and what, in some cases, what the molecules are that lead to it. But there's a logic in the way this process works, because there's only certain kinds of amputations that could occur in nature, right? And so I just kind of keep those things in mind. Some of the stuff we do experimentally is highly unnatural and is never going to occur under normal circumstances, but it helps us understand the process that that does work for the kinds of natural reasons that that loss of limb or tail or whatever might happen. Okay. So the blastema grows at the site of the wound, and the type of structures that are generated, they relate to the position of the cut site, and they are always the more distal structures. So this is the thing to think about. So if we think about the proximal versus the distal limb, and just imagine a simple model of positional information with this sort of maybe one gradient. So there's a signaling molecule, or a ligand for some receptor, a receptor. There's also different kinds of proteins that are present only in activated form in high amounts in certain places, and there's less active form later on, and so on. So just however you think of it, I like to think of it just as a gradient of ligand for signaling, even though that isn't always the case, but just conceptually it makes the most sense. And so along the gradient, you have different levels of whatever this molecule is, and at the highest levels, you're getting the most proximal of the limb structures. In the intermediate levels, you're getting a little bit more distal, and then at the lowest levels, or where there's even maybe zero, you're going to get the most distal structure. So that's the framework that we're using to understand the process. So under normal circumstances or simple experimental manipulation, the limb is amputated cleanly or bitten off, not as cleanly and not as neatly. But so you lose the distal part of it. And then so if that's the case, and you just look at sort of these numbers here. So the gradient here indicates sort of the amount of some kind of morphogen. The numbers are just points along that gradient that are expressions of positional value. So this is a proximal value, this is a distal value, these are intermediate, this is closer to the proximal end, this is closer to the distal end, and so on. So the idea is we would refer to these as positional values, but you can also think of them in terms of the amount of some signal let's say, signaling molecule. So when the limb is regenerated, which it is, and it only takes, I think it's 170 days or less than that. I can't remember off the top of my head. But it's like just a few months to regenerate the limb. And when it does, it regenerates values that are lower than itself. So if you think of this as an amount of signal, then imagine the cells here at position 3, which are the place where the cut site was. They will divide and they can produce more daughter cells. And the daughter cells can either produce the same amount of signal or less, but they can't suddenly start increasing their expression. So that's kind of if you think of that as the principal rule of the process, then it will make a lot of sense. So these guys, when these cells start dividing, they can produce lower amounts of signal, but you can't suddenly get cells sort of up here suddenly increasing and expressing higher levels of that signal, right? That's just not going to. If that happened, then you wouldn't be able to properly regenerate your limb at all. Okay, so this is an example of an unnatural kind of amputation. If you cut sort of here and just remove this part and graft together these two pieces, then the cells that meet each other here have very different levels of signaling. So when I say tell, of course I mean that the cells can detect. I should say just detect, because tell implies consciousness, which, as far as we know, cells don't specifically have that kind of consciousness, but they would be able to detect, like if there's a huge amount of signal relative to this, there's a mismatch, right, between the cells. And so when regeneration occurs, it is, and this has been done experimentally, the limb regenerates properly, and all the intermediate values between 2.5, whatever this is here, and 5, those are regenerated, and we get the proper formation of a limb. And this is referred to as intercalating. So if you are connecting these two things and they don't match, and there's a number of values in between, the generation of those in between values is the process of intercalation. And when that intercalation occurs, the color coding here doesn't mean that the fate of the cells has changed. These are still, the numbers here still indicate that this is the intermediate tissue, the tissue that was missing. The particular bones that were missing are regenerated, and all the muscles and the proper nerves and etc. But to simply point out that it is the cells on the proximal side that divide and produce new cells that then regenerate the structures in between. The cells on the distal side, again, because of this rule, the rule is that when these cells divide, they are not able to produce cells that express more of the signal than themselves. And so therefore they don't divide, they simply remain with their proper already formed distal structures, and the cells on the proximal side are the ones that divide, produce lots of daughter cells. Those daughter cells are then specified properly to produce all the intermediate values. So all the work really of this process comes from the proximal side.